For what it's worth, I wanted to talk a little bit about my experience in the AstraZeneca COVID-19 vaccine clinical trial and talk a little bit about why I decided to volunteer for it, what I learned throughout the process, what the process was like, and more recently, the decision to attempt to deblind myself to see whether I got the vaccine or the placebo control, and the ethical issues surrounding deblinding and blinding in vaccine clinical trials in general. So first of all, why is it that I decided to volunteer? Well, the first and foremost reason is scientific interest. I am a scientist. I have a PhD in chemistry. I have a range of interests. I'm also a teacher. I thought it would be interesting to be able to use this clinical trial as a case study in my course. Also, I am a grant writer. I write many clinical trials as parts of my NIH applications. Now they are usually for wearable types of interventions and not something that goes in the body, but I figured that I wanted to know what it was like on the participant side as well as the principal investigator side. So that is to get inside the process, to be on the other side of inclusion and enrollment reports, biostatistics, and to really have a stake in current events. It was just really exciting to me as a professional scientist. Also, I wanted to teach this in my class on nanomaterials. So there are lots of different chemical engineering and nanoengineering principles that go into the design of a vaccine formulation in addition to the molecular biology. So reasons why a vaccine is or is not stable in cold temperatures or warmer temperatures, why it needs cold temperatures, have a lot to do with the structures of the vesicles in which these vaccines are solubilized. I wanted to have a stake in the fight against the pandemic. I had trouble reading about COVID-19 and, sitting in my closet, doing my work as a knowledge worker. I could do it all from home, and I wasn't really risking anything. Also, since March, my wife, who's a PhD virologist, has been working for operations for one of the three largest manufacturers of COVID tests in the United States. And frankly, I felt a little inadequate, like I wasn't, doing my part. So I knew that if a clinical trial became available, I would at least look into it. So in October at UC San Diego, which is my employer, we got an email inviting us to fill out a questionnaire on our eligibility. And it just asked about, simple questions about medical history and chronic illnesses and medications and so forth. And a little bit about the risk assessment profile too, like how many days per week do you spend at home versus on the job, and how many times are you in crowds of 10 or more people, and so on. And I thought, well, there's no way they're going to pick me because I have a very low risk profile. Although my daughter, who's 1.5, is in daycare, so she's the principal vector into which that could bring the virus into our household. It turns out that our daycare did actually have an infected student and family, and they were shut down for 2 weeks, and I got a test, and a test for an active infection. It was negative, but anyway, in general, I was fairly low risk. I didn't have the risk profile of a frontline worker for sure. So I was basically, when they sent me this questionnaire, there were two categories of individuals that they were interested in. One was, the most obvious case is frontline workers, and primarily people that live in the south part of San Diego County, which are minority neighborhoods. So all of the vaccine study sites were all located south of the Interstate 8. And that makes sense because those are the communities that are putting themselves most at risk doing frontline essential work. The second category were people like me, UCSD, namely UCSD affiliates, people who were not not engaging in a lot of high risk behavior, but had, maybe had interesting, circumstances around their lives, and also because the study was being run by UCSD and had a UCSD principal investigator, so they gave some consideration to whether or not we were UCSD affiliates. So about a week later, I did actually get a call back, or rather an email from the study recruiter, and I had the option of choosing either the Janssen/Johnson & Johnson vaccine or the Oxford/AstraZeneca vaccine. Moderna was also being run at the site, but it was in the last phase of recruiting and might have actually stopped recruiting right around the time that I enrolled. So when thinking about whether I wanted to do, I knew that I wanted to do either Janssen or AstraZeneca rather than one of the mRNA vaccines, just because I really liked the idea scientifically of a viral vector, which, I don't know, it seemed scientifically cooler to me. A horrible reason maybe, but I like the idea of starting with either some non-infective version of a virus. In the case of AstraZeneca, it's actually a chimpanzee adenovirus that has for which humans have no native immunity, and they put the gene for part of the spike protein in the virus. It puts it in your nucleus. It does not go in your genome, but so when your cells divide, it doesn't end up in one of your chromosomes, and then from there you get translation to mRNA and then transcription to transcription to mRNA and then translation to the spike protein antigen. When it came down to Janssen versus AstraZeneca, I was really interested. I was compelled by the fact that AstraZeneca's study design had a 2-to-1 odds of receiving the actual vaccine as opposed to the placebo, which was phosphate buffered saline in the arm run in the phase 3 trial of the United States. So some early trials for AstraZeneca actually used a meningococcal placebo using the same technology but against a different pathogen. So the other reason is that the AstraZeneca site, this is a much lower reason on my priority list, but it was a more favorable location to get to. It wouldn't have taken me as much time, and AstraZeneca seemed a little bit farther along in the approval process, so for those reasons I chose it, but mainly because of the 2-to-1 odds of actually getting the vaccine. It turns out that data from Janssen, Johnson & Johnson are pretty promising, namely because they, in their trial, they actually included a few different antigens, and it has a possibility of longer lasting immunity that doesn't require a booster, although the trial data have not yet been shared. So also the Oxford vaccine was getting a lot of hype. It was one of the first ones to be in the news. It was big news when AstraZeneca decided to partner with them, and I had kind of been following the story on news and podcasts. Interestingly, both Janssen and AstraZeneca had been associated with some fairly severe adverse reactions in the phase 1 and 2 trials, causing both trials to be temporarily suspended in the United States for at least a month each. In the case of AstraZeneca, there were 3 cases of transverse myelitis, which is a serious nerve disease. One of these cases ended up being in the control arm after they deblinded the the investigators to that participant, and another occurred in a patient with a history of multiple sclerosis, which is a risk factor, and ultimately for transverse myelitis, and ultimately there were no data linking the vaccine to adverse reactions in the adverse reaction in the third participant. And it's important to note that in a study that is going to enroll more than 100,000 people worldwide, there are going to be some coincidental adverse reactions, which incidental medical events, which appear to be adverse reactions, but really have nothing to do with the vaccine. Someone could have a heart attack, but they were going to have the heart attack anyway, whether they got the vaccine or not. Just some nuts and bolts about the process. The incentive was $100 in the form of a Vanilla Visa gift card per visit, over 8 visits for 2 years, with most of those visits occurring monthly in the first 4 months of the trial. And that is because that's when they really need to know about safety and efficacy, is at the very beginning. So 2 years is a long time given the seriousness of the illness, and I went into the trial knowing full well that if I were offered an approved vaccine 6 or 8 months later, I would take it, but I would be forced to drop the study. But I think pretty much anyone who enrolled in a study had the same felt the same way, that if a vaccine became available and it were approved, and they were blinded, they didn't know if they actually got it, then they would probably drop the study and take the vaccine once it became available. So I was actually nervous about these adverse reactions. After all, I was intentionally injecting foreign DNA into the nuclei of at least some of my cells, and then it was going to produce an antigen that is a part of the virus that has caused so much destruction and death in the world. But I did read the papers. I read at the time there were 2 peer-reviewed papers, one on a primate model and one on the phase 1, phase 2 safety data in humans, and I decided that it was okay, that it was worth the risk. So I wasn't overly concerned, and in fact, I even felt a little bit selfish about it, because after all, if I got the vaccine successfully, I'd be one of only a few tens of thousands of people in the world who had some degree of immunity, who weren't previously infected. Also, unselfishly, although I wasn't a particularly high-risk individual, I felt that my decision to do the clinical trial held a little more weight, given that I have this sort of public-facing part of my career. Also, I didn't need to do this. I didn't need to put myself at risk at all. I have a very low risk profile. I work in this closet most of the day. I'm an upper middle class white guy with degrees from Harvard and Stanford, who just as easily could have just stayed home in my closet the rest of the pandemic and basically not be threatened by the effects in any direct way. So if I chose to take it, given the fact that I had a really low risk profile, then I think people who are really threatened by COVID, so friends and family members of mine, or maybe people that follow me on Facebook, Twitter, or YouTube, who are essential workers, or who do need to expose themselves to people in the community and risk community spread, that they might be more inclined to believe me because of the credibility of the fact that I didn't need to do this. So nuts and bolts, again. I'd never participated in a clinical trial before. The study location was a bunch of tents in a parking lot adjacent to the City Hall of La Mesa, which is kind of like a sub-city on the outer edge of San Diego, but very much part of San Diego. And the site was run by the San Diego COVID-19 Prevention Network. The people involved were all really nice. However, it was pretty clear that this was a transient operation. Most of the people checking me in seemed like they were learning on the job, and they probably were. And that's sort of to be expected. This is a big job. It requires taking people from a lot of other sort of healthcare adjacent fields to enroll people in the study and do things that they're not used to doing. So there was a physical exam during, so I checked in, I answered questionnaires, several questionnaires about my risk factors and so on. Also, I answered the questions about whether or not I had new loss of taste and smell, had experienced cough, body aches, and so on, several times throughout the visit. And I took a physical exam. I was a little worried that my white coat hypertension, which I've talked about in the episode on mental health, would get me booted from the study because my blood pressure was really high. But after all, I was about to inject an experimental drug into my body. But I convinced them that my doctors knew about my white coat hypertension and not to worry, and they believed me, I guess. So next I went into what looked like a converted school bus that had been sort of retrofitted into converted into like a field hospital. So they took a nasal pharyngeal swab, so the one that goes all the way up your nose for an active COVID infection. Then they took blood to measure background and baseline antibody levels, and then the nurse retrieved a cooler with what looked like a pre-filled syringe of a transparent, colorless liquid. And when she, right in the moment, the moment of no return, right before she plunged it into my arm, and I thought about the 3 patients who had severe nerve disease, neurological illness after getting the vaccine, and I probably suffered a few skipped heartbeats, and a surge of adrenaline when she plunged it into my arm. I usually watch the needle going in, not out of any sadistic tendency, but as an exercise in mindfulness. And so then that was that. The whatever it was in my arm, and I was asked to wait in a tent for 15 minutes to make sure I didn't have an anaphylactic event. So during those 15 minutes, another one of the study workers showed me a wearable device that I would wear to monitor vital functions in the event that I experienced any COVID symptoms, so that they could monitor the course of the disease and see how serious it was compared to the background seriousness out there. So the soreness in my arm took about 5 minutes to settle in. So in my previous life, when we were allowed to travel, I traveled internationally quite a lot, so more than 100,000 miles per year, and I took a lot of vaccines, and some bruise more than others, and this one was maybe somewhat worse than the flu shot, but wasn't didn't leave too much of a visible bruise. So they, I didn't have any serious immediate reaction other than the arm pain, but not something that doing pull-ups for the first time in a long time won't do. Even worse. On the way home, I listened to the song "Fear Inoculum" off the album with the same name by Tool. And the words to the song, "Immunity, long overdue, contagion, I exhale you," sort of seemed especially poignant at the time. So at that point, I didn't know if I had the vaccine or the placebo. So I got the shot around 10 a.m. and it took about 2 hours for the entire visit between check-in and the physicals and the tests. So I was there from about 8 to 10. The vaccine was the last thing that I got, or rather the injection was the last thing that I got. I heard from people who had enrolled in the Moderna trial that the adverse reaction felt like the flu for 24 to 36 hours, except that there were no respiratory symptoms. So I heard it described a little bit like a hangover. So around 6 p.m., I got a little soreness in my joints, which I tend to get when I have a bad cold. I got a slight headache and a very mild upset stomach. My temperature went up by about 1 degree, so it was in the 99s. I took Tylenol and a sleeping pill mostly to make sure that I could sleep through the night in the event that I had more adverse reactions overnight, but honestly, the reaction didn't feel that bad. I was actually completely functional. I could have done my job if it were during the day. Also, as a rule, I'm more paranoid and I'm somewhat more in tune with my body than the average person, I think. So if I thought the pain or discomfort was a 5 out of 10, it was probably would have been perceived as a 2 to most people. So I posted to Twitter and Facebook about the experience and was actually contacted by a reporter from metro.co.uk to share my experiences, and that got published around Thanksgiving, so that was pretty cool for some 15 seconds of fame. And then I didn't think that much about the trial until the booster shot, which was 4 weeks later on day 29. So on that visit, they did a blood test, another blood draw, and a shallow nasal swab, and then the second shot. Then I waited for 15 minutes. They made my third appointment, and since I took that shot later on in the day, like in the afternoon, I didn't develop any adverse reactions before I went to bed, but I did wake up in the middle of the night for about 90 minutes or 2 hours with sort of nondescript unsettledness, but it wasn't that bad. It's possible that I slept through some of the reactions that characterized the first visit. So then the question arose in my mind is whether I wanted to know if I got the vaccine or the placebo. I think that most people, their initial reaction would be, yes, of course I want to know if I got the vaccine or the placebo. But then the scientist in me said, if I know I got the vaccine, would that change my behavior and thus make my data point less useful, or worse, make the efficacy data change the efficacy data, because you're comparing the number of infections in the control arm versus the number of infections in the vaccine arm over some specified period of time. So I think almost anyone but the absolute purest scientist among us would not have at least been curious as to whether or not they got the real deal. So on the other hand, most participants are de facto deblinded because of the sore arm. The phosphate buffered saline solution is really unlikely to cause a week of pain at the injection site, and moreover, that type of pain is not likely to be psychosomatic. Like, you press on it with your finger and it hurts. And in the first couple days, you might have difficulty lifting your arm over your head. But again, not that much worse than the flu shot. So there's really no great way to placebo control a vaccine trial unless you're going to give the placebo arm a different kind of vaccine. The reason that they even try is because they want there to be at least a chance that the vaccine arm and the control arm of the study have the exact same population characteristics, the exact same risk profile, and the same behavior. So if the vaccine arm participants know they got the vaccine, then they might engage in riskier behavior. So in this case, that actually makes the recent results of the Pfizer and Moderna vaccines of 95% effective all the more impressive, because if the vaccine arm participants were going to clubs and going to bars and not wearing masks, then they added more exposure than those in the control arm. So the 95% number is really good. So the selfish reasons in favor of deblinding myself by way of a third third-party antibody test were number one, peace of mind for me and my family. Number two, indulging in my own scientific curiosity, and the kind of scientific knowledge that counts as selfish and not magnanimous. And number three is the feeling that, damn it, I put my body at risk in the face of potentially life-threatening neurological consequences, and AstraZeneca was going to wait 2 years to tell me if I actually got the vaccine. So those are the selfish reasons to deblind myself. But there are serious ethical reasons not to deblind oneself, and there are the reasons that the reasons that I've kind of already talked about are that the control arm and the vaccine arm participants absolutely must have the same behavior, otherwise the efficacy data can't be trusted. So I decided, however, that if I were to deblind myself, I would have to promise that my behavior would not change, not only for the greater good of the integrity of the clinical trial, but also because, and this is really important for everyone who's vaccinated, that at this point, when I'm recording this on January 18th, there is a high probability that vaccinated individuals can still be asymptomatic spreaders. So I've been a double masker in indoor spaces since the early days of the pandemic, and I'm at home all the time anyway, and I fully intend to continue to do so as I had been doing before. So now that I was thinking of doing the unscientific thing and finding an antibody test, there were some reasons to that countervailed against the selfishness and said that there might even be unselfish reasons to deblind myself. So the first is that I had every intention of getting an approved vaccine when it became available, probably by the end of the summer. So I would have to drop the trial anyway after the first five or so visits. If I was deblinded, I could safely give my dose away to the next person in line if my name come up before any of the more at-risk individuals. So they talked about giving the vaccine. So I know in some states they've started giving the vaccine to professors in phase 1B, which in my opinion is I'll leave it there. I mean, it depends on whether or not those locations will require those individuals to be back up speaking in front of classrooms, and if so, then by all means vaccinate them. But if they're just working, teaching class from their computers, then, or from their closets like I have been, then there's no reason to give it to them, right? So if my name came up earlier than frontline workers, than people working at the supermarket, then yeah, I'm going to give up my dose and let someone who really deserves it get it. The second reason is that there's a lot of vaccine hesitancy out there. Like, some huge percentage of the population, even frontline healthcare workers, are nervous about taking the vaccine. So this could be, if I knew that I had it, it would be an opportunity to speak out about my experience and to allay fears that essential workers might have. So if I was vaccinated and I have this platform, then it's my duty to speak out and tell people that it's nothing to worry about. But if I had the placebo, I'd have no basis to do this. Ultimately, I decided that I was going to attempt to deblind myself. I took 2 antibody tests. At first, I tried to get away cheap and do the finger prick lateral flow assay that's available at the pharmacy at Ralph's Supermarket for $25. Now, this was not the right test to take because it tests for 2 kinds of antibody against 2 kinds of antigen. Let me back up a little bit and tell you a little bit about this in case you're not familiar. There are 2 kinds of antibody that it tested for. One is IgM, immunoglobulin M, which appears shortly after infection. It's a multimer of antigen binding domains, and it shows up quickly in the bloodstream. And then IgG, immunoglobulin G, which appears later but lasts longer in the bloodstream. So there were there were also two kinds of antigen in the Ralph's test. One was a recombinant protein of the nucleocapsid protein, which is on the inside of the SARS-CoV-2 particle, a viral particle, and the other was a part of the spike protein, which the virus uses to dock to cells. So the spike protein is the one that most of the vaccines code for, so all of the reagent used in the Ralph's test to detect the antibody for the nucleocapsid antigen, which would have been on the inside of the actual virus, was useless to me. So also, since this was a qualitative colorimetric assay, sort of like a pregnancy test, there was no way to know which antibody would give the dominant signal. So when I took the test, I actually saw faint lines for both IgM and IgG, but the pharmacist assured me that the result was negative, and she told me that she had seen the results of a real positive test a couple of weeks ago, and thus I mentally filed this result into the category of inconclusive. I then shopped around for a more accurate test that fulfilled these two criterion. Only the antibody for the spike protein, and only IgG, since it had now been 4 weeks after the second dose. So Labcorp, which is one of the companies that does a lot of the testing for hospitals, offers a direct-to-consumer test of this type, but it's not necessarily a straightforward manner to figure out which test you're actually buying, and you have to go pretty deep into the technical literature on these tests to figure out which antibody they're testing for and which antigen they are testing for. And this is not something that most people who participated in the clinical trial are likely, this is not knowledge that they're likely to have on board. They're not likely to know where to find it. This is not them. It's just that they're not scientists and they don't live and breathe this stuff. And they also aren't married to a virologist who works in the testing market. So while it's not impossible information to find, it's not readily available. So I ended up going to Labcorp. I actually went there intending to get the quantitative assay, but they didn't offer that to anybody except people with a doctor's order, or to the PIs running the clinical trials. So I got the qualitative IgG for the spike protein antigen test. So sometime overnight, they sent me an email saying that my results were ready, and indeed, I was positive for the spike protein antibody. And while this particular test was associated with a small rate of false positives, I actually tested negative for the antibody back in early spring, when I thought I might have been exposed, and it ended up testing negative, and the test at Ralph's actually said that I probably didn't have the nucleocapsid protein, or maybe suggested that I didn't, and so now I'm 99% sure that I got the actual vaccine, the positive test result for the spike protein antibody, as well as the adverse reactions I experienced after both the prime and the booster shot. So take that for what it's worth. I do not think, as a general rule, people should be deep blinding themselves. I had to go through quite a time-consuming ordeal to do it. It's also not that it's not convenient to take these tests. It took me a very long time of research on the internet to figure out if the test was actually going to give me the information that I wanted, and it requires some amount of domain knowledge to learn what tests are even capable of providing the information you need. So I don't think that there's going to be a deluge of study participants who try to deep blind themselves. It's probably a really tiny number, but in the end, honestly, I'm glad to know that got it. It's not going to affect my behavior in any way. And if I'm offered the vaccine anytime in the next, short while there are still essential workers who are not vaccinated, I'm going to turn it down and give my dose to somebody else. And at any point in the next 6 months, when the AstraZeneca vaccine comes online, and that's the one I'm scheduled to receive, I will turn it down. So somebody will get that benefit. So in the end, I'm glad to know, and also to let the public know through channels like these that the adverse reactions are nothing to worry about, and that a trained scientist like me, I'm not a molecular biologist, but I do know the language of science, and I took the vaccine after I took the shot, even when it was riskier then than it is now, because of the phase 3 clinical trials are almost at a conclusion for AstraZeneca, and after reading the papers and deciding for myself that it was safe. So I hope this story was useful to you, and I look forward to chatting with you again soon. Take care.